but you will need to know who did what to figure out DNA structure and how replication works. So we're ready for part 2. Before we get started on the guys and the processes, let me just check on, this is a throwback to the previous exam where we talked about the cell cycle and cell division, and we talked about how you have to make new DNA to make new cells. During what phase of the cell cycle does DNA get copied or replicated? So you had one piece of DNA and you end up with 2. One chromosome, you make an identical one so that you end up with 2. The DNA gets synthesized during the S phase of the cell cycle, which is a small part of interphase. Okay, so what we're going to be talking about in this next part of chapter 12 are things that happen during interphase, where one piece of DNA gets copied and you end up with 2 identical copies. So that process is called DNA replication, and we'll look at how DNA replication happens, and we'll look at how we know what happens and who figured out what happens and showed how replication works. One thing we know about replication that was proposed by Watson and Crick and confirmed within a few years of their publications is that DNA is replicated through a process that is semi-conservative. There's a pair of guys who figured this out or showed that this is in fact how replication works, and you'll need to know their names. We'll get to them. But what they figured out, what they showed, is that when DNA is copied, the original double helix is shown here with the blue backbone. So here's the original. In order to replicate that DNA, the original strands get separated from each other. The hydrogen bonds holding the base pairs together break. And then each of those original blue strands gets used as a pattern to build a new strand, which is shown with the red backbone. And this new strand has nucleotides that are hooked together following the base pairing rules. So where the old strand has an A, the new DNA that's being made will have a T. And where the old strand has a G, the new strand that's being made will have a C attached to that newly made growing strand. So both of those strands of the original double helix are used as a pattern to make a new strand shown in red. When we say this process is semi-conservative, we mean that the new DNAs that we get look like this, where there's one of the original strands, one half of this DNA is the original strand, and that is paired with a newly made strand. It's conservative in that you still have the old strand, but it's only semi-conservative because in addition to the old strand, there's a brand new one. The process of sticking together these A's, G's, C's, and T's together to make the new red backbone strands is primarily done by an enzyme named DNA polymerase. It makes a new chain or a new polymer of DNA. DNA polymerase can't build DNA all by itself. It does have a lot of helper molecules, some of which are also enzymes that participate in the process. But the one that's actually hooking together the A's, G's, C's, and T's with the phosphodiester bonds, that is DNA polymerase. Something else we know about replication is that a piece of DNA doesn't just start getting copied anywhere. You need a special sequence of bases, special string of A's, G's, C's, and T's in order for replication to begin. So that start sequence that's required to begin replication is referred to as an origin that's short for origin of replication. A eukaryotic chromosome, which will be a very long piece of DNA, will typically have several origins, and replication starts at each of them, and they kind of, the replication process kind of gets coordinated when the replication from one origin and the replication from another origin kind of get close to each other. Prokaryotic chromosomes are just a single circle of DNA, and they're not as big. And a prokaryotic DNA will typically just have a single origin of replication per molecule. I say typically, I don't know of an exception to that. So always, almost always. In order for DNA replication to begin at the origin, you have to have some molecules that can start pulling apart the double helix at the origin. And this enzyme named helicase can take that double helix and helps to pull those two strands apart, breaks the hydrogen bonds that were holding the A, T, and GC base pairs together. And then some spacer proteins that are called SSBs will help, once they're separated, it helps hold the two strands apart so they don't snap back together. When the two strands get pulled apart, it actually creates some strain on the double helix over here and over here, especially if it's a circular prokaryotic molecule. So to help kind of keep this part of the molecule from getting bent till it almost breaks, there are these enzymes called topoisomerases that will kind of help handle the pressure of having this part of the double helix becoming single stranded. They kind of relax the pressure a little bit. Okay, so we got the helicase enzyme that can pull the strands apart. The SSB protein shown in green help keep it apart. And we have kind of a bubble here where the double helix has separated into two single-stranded areas. So this is sometimes referred to as the replication bubble. And with an electron microscope, you can actually see it. It's that big. DNA copying will happen over here and over here. So from one origin, you actually get replication moving in both directions away from the origin. We refer to that as bidirectional replication, and that's typical for chromosomes of cells. Okay, so our topoisomerase prevents the double helix from getting tangled up, from having this part pulled apart. And then we have our replication bubble, and this shows replication happening over here on the left and replication happening over here on the right. So it's bidirectional replication because this one origin fires both to the left and to the right. And those regions on each end of the bubble where the replication is happening, right here, those are called replication forks. So this is a replication fork. This is a replication fork. We'll spend some more time looking at this picture. But replication is going to come from the origin and head over toward the left. And it's going to, once this part gets replicated, this part of the double helix will open up and the process will continue to push forward into the double helix until the whole entire molecule gets replicated. The original strand is shown with the light blue backbones here, and then the newly made DNA is shown with a dark blue backbone. The little red bits, we'll get to what those little red bits are in a minute. Okay, so we got our replication forks where the arrows are pointing. So I said I'd get back to the little red bits. And one thing you should notice here is we've got more little red bits and less of the dark blue stuff on the top strand over here on the left replication fork. And on the bottom strand, we had just one of the little red bits and then a whole lot of blue. So there's a difference in replication of the two strands at a replication fork. One of the strands can be made in a long piece, whereas the other strand is replicated in short little pieces. They're going to have to get hooked together afterwards. And the reason for that is that it's a property of that enzyme DNA polymerase. It hooks the A's, C's, G's, and T's together to build the new strand. It's making this dark blue new DNA. The thing is, DNA polymerase can't just start making a new piece of DNA from scratch. It can take a little string of nucleotides and add bases to it, but you already have to have the string there. So that little string we call a primer, and that's what's represented in red here, here, and here. So that little starter DNA, or it's not DNA, the starter sequence or primer, it's actually not DNA, it's RNA. So there's this enzyme named primase that can build RNA from scratch. So it makes a short little string of RNA pairing with the DNA, and then the DNA polymerase can add on to the rest of that. So how come we have just one primer and a really long piece of DNA on the bottom strand at this left-hand fork, and we have these shorter pieces, each of which has its own little primer and a little bit added onto on the top strand at the left-hand replication fork. And by the way, if you notice, the right-hand fork is reversed, where the top strand's the one where you have the one long piece, and the bottom strand on the right-hand fork is where you have the short pieces. Okay, so we talked about primase. Primase is making the red bits, the short little RNAs the DNA polymerase can add on to. But then why, on one strand, why do you just need one little primer, and then the other strand you make a little bit and then you need a new primer to make more? It's another property, another limitation of DNA polymerase. Okay, so there's a difference between the replication on the top and on the bottom strands, and we refer to those two strands as being the leading strand and the lagging strand of replication. So at any replication fork, one strand will be the leading strand made in one long piece, the bottom strand on the left, the top strand on the right. And then the other strand is the lagging strand that gets made in short little pieces. The fact that that one strand has to be replicated in short little pieces, we refer to that as discontinuous replication. And those short little pieces that are made are referred to as Okazaki fragments, because Dr. Okazaki, the doctors Okazaki, figured out, demonstrated that these are produced. The reason the lagging strand has to be produced in these short Okazaki fragments that are maybe 700 to 1,000-ish bases long is because the replication fork over here, the replication fork is moving to the left. And so the copying as a whole is moving to the left, but the DNA polymerase enzyme can only build DNA in the 5' to 3' direction. Okay, so not a problem on the bottom strand here, because the bottom strand, the new DNA being made 5' to 3', after this part gets made, when the double helix over here opens up, you're going to be able to just add on and continue to grow to the 3' direction going to the left. But on the top strand, remember that the two backbones of the two strands are anti-parallel, 5' to 3' going in opposite directions. So on the top strand, the DNA polymerase has to move from left to right if it's going to be building DNA in the 5' to 3' direction on the top strand. So the enzyme has to go left to right, but the double helix is going to be opening up over on the left. So the only way for that to work is for DNA polymerase to make a little string of DNA, and then when the replication fork opens up more, the enzyme jumps backwards and copies another piece of the DNA. And as the replication fork opens up again, it's going to have to jump backwards and just keep going backwards and copying backwards and copying, jumping like that, and making those little Okazaki fragments. They're going to have to be joined up eventually, though. You can't leave the RNA primers there, and you can't leave this as a bunch of separate little strings. They have to be hooked together. So this is another picture that shows leading strand and lagging strand replication. The leading strand is up on the left, on the top, sorry. And then the lagging strand, we see our little Okazaki fragments here. And here the RNA primers are the kind of turquoise color. What has to happen is the primer has to get chewed off and replaced with DNA. And the nucleotide subunits are really similar for DNA and RNA, but they're not identical. So you have to take off the RNA ones, and then a DNA polymerase can add on DNA ones that correspond with the complementarity rules. And then you're going to have short little pieces of all DNA, but there's still going to be a gap between the strands. Between this Okazaki fragment and the next Okazaki fragment, there's a gap. You need a covalent bond to tie those two pieces together. And that, oops, sorry, and that gap is filled in by the enzyme DNA ligase. Ligase means tie. So DNA ligase ties together the strands of the individual Okazaki fragments to make a continuous string of DNA. Now this lagging synthesis replication, it's more complicated, so it takes longer than the leading strand replication. And that's where lagging strand synthesis got its name. The fact that it's slower, it lags behind the replication of the other strand. Okay, so here's a picture that shows, yeah, DNA ligase will quickly take those Okazaki fragments once the primers have been cut off and replaced. The ligase will hook together, make a covalent bond between the Okazaki fragments, and create a continuous string. So makes a new phosphodiester bond right here to put together those Okazaki fragments.